Hi, and welcome to Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Okay, here's another video on meiosis. I know, there's lots of them on the web out there, and lots of really good ones too, and I hope mine is an okay one. But this is my second part on my series on sexual reproduction and inheritance. And what I want to talk about today is how do you get 4 haploid daughter cells from a diploid parent cell? And then I want to talk about the origins of sperm cells and eggs. And I'm not talking about their evolutionary origins. I'm talking about how that happened in a human today, because it's kind of interesting. And we're really different as men and women when it comes to the way that our bodies form our gametes. Even though meiosis is still the same, our bodies do things really differently, which we'll cover in gametogenesis. Uh-oh, did I use a big word again? Gametogenesis. Gamete, that's your sperm and your egg. Genesis means origins of. So when we talk about gametogenesis, what I'm talking about is the formation of sperm and egg. But let's talk about this. Let's get into the nuts and bolts here of how we're going to generate 4 haploid daughter cells. I used another word, didn't I? Haploid. Okay, we got some terminology. Always remember, diploid, di means 2, ploidy having to do with chromosome number. So if you're diploid, you've got 2 copies of your genome. And if you're haploid, you have 1 copy of it. So our somatic cells, the cells of our body, we're diploid. We have 46 chromosomes. You've got 23 from mom and 23 from dad. And our gametes are haploid, which means they only have 23 chromosomes. So how do you go from 46 to 23? Let's talk about that. Well, first of all, you have 2 rounds of cell division, which is very different from mitosis, which only has 1 round of cell division, but that's also asexual reproduction. So in meiosis 1, this is interesting. That's when the daughter cells actually go from being diploid to haploid, and we'll talk about that. And then in meiosis 2, that's when the sister chromatids are pulled apart. Let's take a closer look. So we know that meiosis is going to generate 4 different daughter cells. Notice I said different. They're not all the same. And it's these events in meiosis that make them different. And that's going to be the subject of my next lecture. We talk about crossing over and independent assortment and random fertilization, or semi-random actually. So in sexual reproduction, we know that we're combining gametes, 2 haploid gametes, into a zygote, and that forms an adult. So in sexual reproduction, we're taking 2 gametes and we're fusing them together to form a new organism. And those gametes, of course, are haploid. So in humans, they're each carrying 23 chromosomes. And those gametes fuse together to form a zygote. And the zygote, of course, is diploid with 46 chromosomes. And we would say that 22 pairs of those are homologous chromosomes. And that the last pair, the 23rd pair, which are the X and the Y chromosomes, that's our sex chromosomes. And if you're a man, like me, if you're male, they're not homologous chromosomes. The X and the Y chromosomes actually carry different genes. So in meiosis, let's start talking about the steps here. One key difference is if you're doing mitosis, you copy your DNA. You do the same thing in meiosis. And then what happens is the 2 copies of the chromosomes remain together to form the sister chromatids. Now remember, when they're connected, the sister chromatids are still a single chromosome. But let's say we've got 23 pairs of chromosomes. That means I've got chromosome number 1 from mom and chromosome number 2 from dad. And when they replicate, they form, how am I doing this? They form the 2, these are my sister chromatids for chromosome 1 and chromosome 2. Okay. Well, in mitosis, I just line them up and then pull them apart. But in meiosis, they're going to come together and form the structure called a tetrad. Tetra means 4. So I've got chromosome number 1 from mom, chromosome number 2 from dad. They come together and form the tetrad. So remember, these are homologous chromosomes. They all have the same genes in the same location, but you might have different versions of those genes that you get from your parents. Those are those alleles I keep talking about. So we line the tetrads up in meiosis 1. We line them right up in the middle of the cell, just like you would during mitosis. And then when you switch to anaphase, remember you've got prophase, prometaphase, metaphase, and anaphase. When you go to anaphase 1 for meiosis 1, we got a tetrad, and we're going to pull apart the homologous chromosomes. And because I'm pulling apart the homologous chromosomes, the cell becomes haploid, even though the daughter cells still might have 46 sister chromatids. Because what happens now in meiosis 2, I've got all my sister chromatids, I'm going to pull them apart into the daughter cells. And now each of my daughter cells are going to have 23 chromosomes, but they might have some different combination of chromosomes from your mom and your dad, which is one of the reasons why they are different. I'll go into that more in my next lecture. But the idea here is through 2 rounds of cell division, after meiosis 1, I become haploid. And then at the end of meiosis 2, we pull apart the sister chromatids. We have 4 unique daughter cells. Okay, let's start with spermatogenesis. Sperm means like seed, and spermatogenesis means the origins of the sperm. Now, of course, in men, this takes place in our testes, and it does for all mammals, actually, and begins around the time of sexual maturity. For us, it's adolescence. And you begin with a self-generating stem cell, right? Now, a stem cell is just undifferentiated. It's not expressing particular genes to make it become a cell type. So they're undifferentiated. They can differentiate into other cells. You begin with your primary spermatocyte. All right, cyte is just cell, and it is diploid. It is 2N. And what it does is it's going to go through mitosis. Yes, I said that correctly. It's going to go through mitosis. And it's going to form 2 daughter cells that are both diploid. One is going to remain a primary spermatocyte. The other one will then commit to meiosis. Then my secondary spermatocyte, it will begin to undergo meiosis. And of course, it becomes haploid after meiosis 1. And then it will go through meiosis 2 and form our 4 haploid gametes. And then there's a maturation process where basically the cell sheds most of its cytoplasm. And then the head of it just is basically the capsule of the nucleus containing all the nuclear material, the chromosomes. And then it grows a tail. And this takes a few days for that to happen. And the thing here is that all 4 of these daughter cells are viable sperm. And we, as a man, we produce on average about 3,000 sperm a second every day of our lives once we become an adult, unless something happens. Next, we have oogenesis. This, of course, is the formation of eggs in females, and it's quite different than in males. Now, there are some similarities, and one of them is that it begins with a stem-like cell that reproduces through mitosis. It's reproducing asexually, forming many different primary oocytes that are diploid. Now, here's where things start to diverge rather quickly. Okay, so first of all, this takes a lot longer and can be halted for decades in females. So starting about 7 months in development, I mean, when you're talking about the fetus being only 7 months old, this is when we start to get the primary oocytes are being formed, and they're being halted in prophase 1. That's interesting, right? They actually begin meiosis before the child is even born, but it's halted. And of course, this takes place in the ovaries. So once a child is born, they basically have all of their eggs. And you might form millions of primary oocytes, but by birth, you only have about 400 or 500 of these primary oocytes that are going to be viable. So another thing, not only does gamete formation begin in the womb, by the time the child is born, she most likely has all the eggs she will have for the rest of her life. Now, there's some indication that that's not completely the case, that sometimes women actually do start producing eggs, but we're not sure. That we don't know yet. So once you reach adolescence, once you begin puberty, about every 28 days, you ovulate. And what happens is that is that the primary oocyte that's been arrested in prophase 1 will then undergo meiosis 1. It will complete it. Now, the secondary oocyte, after completing meiosis 1 upon ovulation, will be halted in meiosis 2 unless it becomes fertilized. Now, the meiosis in eggs is completely unequal. You're not producing equal daughter cells. In fact, you produce what is called a polar body. This is a tiny little thing. You see, the primary and secondary oocytes, they're going to maintain, one cell is going to maintain almost all of the cytoplasm and organelles. And then the polar body is just this tiny little thing to shed the excess genes. So in meiosis 1, you're getting rid of your haploid chromosomes. And then in meiosis 2, you're getting rid of your other sister chromatids. And meiosis 2 only is formed after fertilization. So these polar bodies, like I said, they're just reducing chromosome number. There's some implications here, right? If you're 35 years old, you've had your primary oocytes have been halted in prophase 1 for 35 years, okay? Now, these primary oocytes can actually be maintained for up to 50 years. But you can imagine something that's been arrested in a state for so long, things might start to happen. So even though we both have diploid stem cells giving rise to our primary oocytes and spermatocytes, even though we both have diploid stem cells giving rise to our primary oocytes and spermatocytes, men, we continually produce ours from adolescence onward. Women do it all when they're young, and then they're maintained for decades. And of course, this leads to problems. One of them is called a nondisjunction. Now, that's a fancy way of saying that you don't separate your chromosomes equally during meiosis. See, if you actually produce a gamete that has extra chromosomes or too few chromosomes, that's called a nondisjunction. And one of the most famous ones of this unequal separation of chromosomes, of course, is trisomy 21. Most nondisjunctions are fatal. So if you don't get a chromosome number 1, the embryo dies, the zygote dies. It'll undergo a few rounds of division, and that's it. And in fact, many miscarriages, not all, but a lot of miscarriages are due to chromosomal abnormalities due to a nondisjunction. And after about 35 years of age, the rate of nondisjunction becomes higher. Now, chromosome 21 or a trisomy 21 causes Down syndrome. That's where you've got 3 copies of chromosome 21. And it's viable. I mean, you do produce children with Down syndrome, and they can range from mild symptoms to quite severe. It's one of the few trisomies that can actually survive. Okay, well, I hope you enjoyed this video on how meiosis works. This has been another episode of Tom Kennedy Science. And next, stay tuned because we're going to learn how meiosis generates genetic diversity.